We must reach the top. Yeah, our maps indicate that when we reach the top of this edge, it should flatten out considerably. And one of the things we want to do is really compare who's living on the edge and who's living on the platform portion of this, one of the many step terrace-like features of the edge of the Blake Plateau. We're on the edge called the Blake Escarpment. And all of these slabs of rock used to be...
Pilot, this is Watch.
Yeah, go ahead.
Can we zoom real quick on the coral that's just below center left now?
Center left.
It's like stairs, kind of.
Yeah, this one right here.
Yeah, that's the one.
Video, let's go for it. Get closer to it as we go. Kill lasers, go pilot.
It's definitely a black coral. That's good, Pilot. I think that's enough for someone to get an ID on it, but we should keep going.
Copy
Thank you.
All right, video come wide. Get lasers back on.
And it looks like we're back into this
Scott says possibly parantipathes or taxipathes.
Rich area.
Those look like steps and things, yeah.
Yep.
Yeah. So each of these steps is a layer of rock. Go ahead, Stephanie. Oh, look on the right. Yeah. Beautiful.
Pilot's Watch. Can we look at that big fan on the right?
Copy that.
Thank you.
And there's something down here, too.
Wow.
I tried to keep moving, but...
It's really hard.
But these step features are really cool.
Yeah.
I think that's an accurate description.
Candelabra. Oh, that is
beautiful. That is awesome.
Ooh.
Definitely have not seen one of those. I believe this whole, well, at least today.
I'm surprised it's something.
So this is going to be a bamboo coral. And this is in a new species, new genus that Les Watling and I have been working on describing. And I'm a bit frustrated that we haven't finished it yet. That's our own fault. But let's get a closer look and I'll tell you some more.
Kill lasers.
Great, thanks, Scott. I do understand that describing species takes some time, so.
All right, video, if you want to come in.
Yeah, so it's got this classic candelabra-type shape. And when this is a small coral, younger, it grows as a trident. So you see it's got that main axis, and then it would have 3 upright branches, so it just looks like Neptune's trident. And then as it grows older, it begins to put out more of those upright branches, kind of adding them on to the edges, so you get this really big candelabra-type shape. And we first saw this growing on the New England seamounts on a cruise back in, I think, 2005 or so. The New England seamounts are a chain of underwater volcanoes that are several hundred miles north of here, basically off the coast of Massachusetts. And this was the first octocoral, deep-sea octocoral, to have its mitochondrial genome sequenced. We did that in our lab with Mercer Brugler. And that's showed us some really interesting things, like, for example, that some octocorals have a different mitochondrial gene order, and that had been the first time that had been shown among the octocorals. So a lot of interesting biology has come out of the early collections of this thing, and we've now seen this in both the Pacific and the Atlantic. So it appears to have a pretty wide distribution.
Scott, this is Leslie. What kind of age are we looking at for this? It's a fairly large specimen, or what do you think?
That's a really good question, Leslie, and a really hard one.
Yeah, I'm sure it is.
This particular species hasn't ever been aged before. What you would need to do is have a sample, particularly of that lower, the main part of the axis, and basically do some geochemistry on the skeleton going from the interior to the exterior. But I agree with you, based on the typical growth rates of species in the deep sea, this must be really old to have this many side branches. It's a particularly large one.
Yeah,
I've seen large ones like this before down in the Bahamas
does that go for lasers,
region,
or do you want to keep them on
to
for
our
this?
south. So we know they exist, but I would guess this is more than 100 years old. maybe more than a couple hundred years old.
Yeah, I figured it was probably quite old because of its size, and one of the reasons I asked was actually a geologic reason. Is because it is sitting on what looks like a very tenuously positioned slab, but if that coral has been happy there for many decades, that tells us something about these slabs. They're not just sliding down the slope day after day. So it's kind of,
How is it looking over
I think
there?
these slabs are almost cemented in place maybe
So
by
it takes a few
some
seconds
of
to
the
boot up.
sediments
Is that what it
maybe.
I don't know, I'm asking.
Who knows, but
it might be
I
doing
wouldn't
some
have thought
auto-adjusting.
that this area would have
It looks better now,
stable
but you're a
slabs
little bit
like
low.
this.
Whoa.
Unless
Yeah, that's
it's
a really good observation.
unless it's good at surfing.
We can use biology to inform geology and geology to inform
Yeah,
biology.
yeah.
Yeah, if you wanted to do still, which don't feel the need to, but be up
That
and
is
closer.
a beautiful
and
specimen,
closer?
isn't it?
Maybe
To
just
fill it? Do we
Yeah.
have to almost back up?
So Les Watling from the University of Hawaii
This is
and
really
University
dark.
of Maine and I
Yeah,
were
it's a little
remarking
cool.
in the
There's some
chat room
studies we need to
earlier that a
address.
lot of the observations that we've been making
like once
today
we
of
get to
some of the
surface.
communities, the
Studies.
sponges and the corals and so on,
Like on actual
are
camera.
reminding us a lot
Yeah,
of
to
these
the
offshore
settings. I know we have a new
volcanoes
lens.
further to the north. And
That's
that's really
just the work
interesting
we
because
need
it
to
tells
see
us
what they
something
want.
about the
Copy.
connectivity, the
Good
ability
enough.
of larvae possibly to disperse offshore to those seamounts or from the seamounts to this coast. If we look at some of the canyons further north
yeah,
between
so
here and there, we don't see some of
come to
these
center,
organisms.
right
so
there.
it's an interesting connection. And we saw something
Video
similar down
again.
in the Bahamas. So there is some kind of a connection, but it's not complete along the continental slope.
Video, go for zoom.
We're going to zoom into the junction here, and also it gives a really good sense also why these are called bamboo corals. You can see those segments with the dark bands. And Scott, are those
And you can
episodes
see,
of
one the
growth?
things that we look for when we're describing and identifying bamboo corals is where the branches
Okay,
arise
video,
with
let me know when
respect
you
to
want those
dark
lights
bands.
on.
And this is a really nice image showing that the branching is coming out of that node, the dark part. And so this is what we would call a nodal brancher. An internodal brancher is the branches coming out of the white part.
And these nodes, do they occur after a certain length of growth, of time of growth?
So that's a really good
So for now,
question,
maybe
and
see your
that
sonar?
seems to vary among species, but we don't have
wow.
enough data, hasn't
That's,
been studied that clearly well enough to
downhill
know
after
for
that, or you
certain.
think that's
But there are
There's
some
supposed to be
taxa
a
where, yes, it does
saddle at
appear
some
like
point, right?
they can
Yeah.
have a longer
according to
internode before a
calculations,
new
it's
node is
25
put
meters
in.
away.
Some others, it appears to be shorter, but we haven't had
This
the
is
ability to really do any
25.
good
So could there
experimental
be a
work
map offset?
to
and
see
that could
how
be
that's
the
impacted.
top of the hill?
Yeah, it's too bad
Or
we
it could be like a
can't
small
use them
cloud.
as
is
mostly
what I'm guessing.
protein, and the internode is mostly
So we'll
calcium
see.
carbonate. And that was a nice view. You could see that
So we've
trident
already got you at
appearance. So you can imagine
1260
when
right
this thing
now.
was
And
young,
that's what I had the
before it had
top
a chance
as,
to put out all these
1260.
other branches, it would have looked like a three-branch pitchfork.
Huh.
And actually, earlier than that, it may have only been a whip. And this is one of the interesting things in trying to identify
Actually,
these.
is that
They're
good?
not growing from the base.
Any more
they're going
views
from
on
the
this
tip. So presumably
subject?
it grew unbranched until some signal to begin that trifurcation.
Copy. Video, is there anything else you want? Oh, okay, great. Are you happy, video? Did you get great? Then I'm also happy. So is Nav and Copilot. We're gonna
Nav's
move
also
on.
happy.
That's absolutely beautiful.
Do you want the...
We're all happy here.
Okay. Killing the fog
Cool.
lights. Big difference right there.
Yeah.
All right.
All right, let's keep going up.
Okay, so I'm facing 280.
And we're still seeking a rock
Yeah, it's
sample that we might want to try again.
You want to move? You're looking at me like you want to move.
Now that we know
Hold on.
these
They're talking about a sample.
probably carbonate rocks
get
are
lights
quite
real
friable,
quick?
maybe we can just
Or I'm sorry, lasers. Sorry.
scoop it with
Quick, turn
the
all the lights off.
manipulator
Is
arm.
this something you want to try for?
Can we get a quick zoom on it?
Yeah, videos.
It doesn't.
Snap it.
Looks a little.
That might not be what. That might not be what you want.
Nah, I'm
Just
looking
do
more for the
manganese.
bright white.
Yeah.
That's come from above.
Come wide, video.
It's one of those solitary corals growing on. it.
Yeah.
Yeah.
Pathy's here on the left. Sticker Pathy's in the
We have
center.
Cheryl back from lunch. We appreciate
not even gonna
Stephanie
ask what I
filling
missed.
in. No, we're not going to tell you. You're just going
Good.
to have to wait.
It was torture to step away.
Yeah, if they don't want a sample, then we can go ahead and get a move in.
I don't know what
Look at this.
that.
Ooh, there's a
pink
prime suspect rock over there when we
To
to the right,
to the right.
just out of the field. If maybe you just use the tongs to cradle it
That is a
underneath
big
it.
one.
That's a big one. Oh yeah, it
It's
is.
about half a
Yeah,
meter.
it's pretty large.
Oh, it looked so tiny.
ROV nav bridge, we are stationary at this time. Not sure if I got that message to you earlier. Over.
Copy that. Move complete.
It's a sea
Thanks,
star.
Fred.
Maybe we could do
Maybe
a
an Anthomastus.
quick zoom on that.
Yeah, video snaps sea star.
I think we've passed several of these. We haven't done a zoom as far as
All
I
right.
know.
I might do a move then. Or maybe just wait just one more second. See if I see a rock.
Lots and lots of.
Yeah, there's
Dead
a lot of
Selenisphilia.
little.
Options up there.
Yeah.
Okay.
Little red eye spots at the end of the rays or the arms.
That looks really cool from this camera. Looks like an Asian.
Oops,
Asian.
Video back. There we go.
Yeah, it's amazing how much of the dead coral, it's so blackened. It's been there a while. See a nice edge of this, what is likely limestone. Very friable. It's more like heavily indurated muds, very compact muds. We tried to pick up a rock sample of this earlier and it just disintegrated in the manipulator arms.
All right, great. Could probably get lasers back.
Manipulator.
So as a reference point, Scott France was talking about collecting these bamboo corals in the Bahamas escarpment, and it turns out that they were collected at a similar depth range between
What's that purple
1,255
thing
and
on the left?
1,468
Purple on left.
meters or so. So right in the depth range that we're seeing them today. But I don't think any of us expected that there would be so many here, and it does make for a good stepping stone from the south to the north.
Yeah, everything black in this image is encrusted coral.
Use
Coral.
Yeah.
informing
It's
me
dead
of
coral
the
Dead
skeleton.
coral.
deepest
It's incredible.
water transect.
Makes up quite a habitat.
It
That's
makes
a
you wonder
beautiful spot.
why
Previously was 800 meters depth.
why it
and
was
now we
here and
want,
why it's not here
950
now
meters.
in that
Copy.
Changes the
abundance.
alpha time by
From my experience, we're going to
10
find a
minutes.
lot more
Video,
live
come
when
on
we
in.
get to the top, and it rolls down as it dies.
So this is just debris.
coral
Okay.
debris.
So I guess that's the same type of sponge or something pink living inside of one of those.
Yeah, we were seeing some scale worms inside
Might
them
well be
earlier.
that, yeah.
Levi, are you guys giving also half an hour
We
like the
saw
other day?
a yellowish
end?
green
Okay.
variety of the same thing.
Although
Yes.
Today might not be needed. 'cause
Yeah, we're
it's at
good,
the wind.
thanks.
Great. Video, if you're happy, we can come wide.
Okay, so if Sean keeps moving, I feel like we could get a move in.
Copy.
It's about
Sean agrees.
20. A 20-meter move would put me right on top of him.
Wow.
Bridge is RV nav.
Bridge over.
More sponges.
Yeah, we'd like to
more
move same as last.
Stored
20
pathies,
meters,
black
bearing
corals,
280
more
degrees,
Selenisphilia.
speed decimal 2.
Excellent. We will do 20 meters, 280 degrees, speed decimal 2.
Good copy.
More stichopathes.
So you don't think the Selenisphilia lived here? Because it really
These all
seems like it's coating
animals, Sean?
rather than being piled up.
saying, yeah.
It could be. I mean, it's definitely living on some of the.
ROV nav bridge,
Lesser
move
slope.
initiated.
Yeah.
Move has
There
been
it is.
initiated.
And it
Thank
looks
you,
like
Bridge.
it's attached as opposed to just
But isn't
resting
it
on.
possible that this was living here, thousands of years ago?
Copy.
Yeah,
What's
yeah,
that?
that's why
That's
I'm wondering because it's
hard to see
so
how close it is.
overcoated.
That's, I think what we see in the New
Maybe it's not
England
that
seamount
close.
evidence that
Better
at
safe
some
than
time
sorry.
ago the environment was
Does
somewhat
get nice
different.
steep there, too.
They died, but
So
nothing
I'm gonna
very-
move the lower swing arm a little bit. to
Yeah,
see
and
if
that's
it
why I
fixes
was
that
wondering
Yeah.
what would have been different,
Go
what
for it.
conditions were different here when they lived
And this
that
slope
no
is,
longer exist.
Actually, it's
This
gone
slope
now,
is awfully
right?
steep for them to just be laying there too.
Is it?
Yeah, yeah.
There's that might
This
be
is great.
on that. Oh, we're getting to some ledges now, and that
Okay,
might be.
so I'm moving the lower swing arm video so
I don't
that
think
we
we
know.
expected
the
vertical ledges
flare might
anywhere.
need a-
Yeah.
Like a reflection.
That looks like a
Froth
Selenisphilia
or
there on
white
the hanging off the
rock,
bottom of that
maybe?
ledge.
Possible.
Sorry, Washley, did you call for something?
And often there's desmophyllum that grow.
Do you want to zoom in on something, Cheryl?
Yeah, Pilot, if we could zoom in on the coral that's hanging down from this ledge, please. Thank you.
Yeah, the one coming right there?
Yeah.
Great. Video, come on in. You can kill lasers real quick.
Yeah, so it doesn't look like there's any of that that's alive as far as the stony coral, but it's being inhabited by lots of other things, hydroids.
But perhaps some of it's a little more recent because it's less encrusted.
Yeah. That's great. Thank you, Pilot. If we could take a look to the left, there is a black coral there.
Copy that.
Yeah, there we go.
And you may have described the type of sponge underneath it earlier when I was away. Are these characteristically shelf types of sponges, or?
I actually haven't seen this.
Oh, you haven't? Okay,
before, so
maybe we
I'm
need
not
to take a
quite
peek
sure.
at it. Yeah, I've seen a few of them.
Rising a little. There we go.
See, it has this little Galatheid on it, a Europtychus most likely.
And Tom, Hands Connect says that the yellow sponge was growing as a bracket fungi. Taking advantage of that hard substrate, but sticking out farther into the current.
All right, video, let's go for sponge. Actually, video, come partial. I'm going to kind of drift it a little bit.
Parantopathes, Tina
Get closer and get
comments.
some more light.
Yeah.
That's good.
I
right
believe
there.
she's referring to the sponge.
No, she's referring to the
Oh, I'm
black
sorry.
coral that's sticking up
Shows you
there.
how much I know.
And you can see
All right, video, you can come back in.
dead cup corals
Yellow
here.
sponge looks pretty different from some of the other things that we've been seeing before.
Yeah, that's what I
Recognize
thought.
that morphology.
It's too bad we don't have Shirley Pomponi joining us today.
Yeah, it's almost like there's kind of a crust on the outside that's a somewhat different color, and then the yellow sponge inside. And
That
you know,
moves
it
complete, guys.
reminds me of
Okay.
the boring
ship-wise,
sponge
serious
Cleona
point,
that
single bit.
Yeah.
infest oyster shells, for example.
All right, video, you can come on.
It's like there's some sediment on the surface. And then to the left of that sponge, you see those cup corals. Those were most likely Desmophyllum dianthus. They didn't look too
We'll keep an eye
lively
on
There's
though.
that.
several of
Serious
those.
sonar.
Oh my gosh,
guys.
Yeah, I
there's
thought
bamboo
I-
coral.
Not sure how high that wall
Oh,
is.
Got
look
a
at that.
really
It's beautiful.
beautiful
Copy.
bamboo coral on
Should we
the
take a
left
peek?
here. It looks like it took a right-hand
Right
turn.
angle
You see it. I think you
ROV
see
now.
it.
You see
Yeah.
that, Scott? Maybe it hasn't
That's
reached it
probably
yet,
it.
but
Yeah, right.
interesting
Yeah,
growth
it's
for
Complete.
it.
Thanks, Bridge.
Yeah, I think
Looks
the line
like the
that we
larva
were
settled,
seeing
started
was
to grow and said, oh, it might be a better
That
idea if I
line
go
right there.
upwards
Yeah,
instead
that's
of
what you-
sideways.
Oh, it looks like we got
This
a...
is
This
a
is
different
gorgeous.
species from what we've
My
seen
gosh.
earlier today. I'm guessing this might be considered
Wow,
keratoisis.
this is quite a shelf.
Yeah.
We
Be a
need to get a
shelf of
closer
life.
look.
We got a nice fish up there on the upper left
fish.
ledge too.
Upper
I'll
left.
make sure we get the fish before he swims out of the picture.
Video, go for fish. Nice.
Oh, that's a beauty.
Wow.
Look at him. What coloration.
Fully expect when we get to the top of this ledge to see lots of Bursinges as well. That's the typical thing you see. Oh, nice. Maybe
The
some
lights.
codling here.
The fog lights probably would illuminate that,
Huh.
wouldn't it?
I
Great
don't remember
habitat.
seeing any in
Do we
this
kill
color
lasers?
before.
That's a good shot.
That's just beautiful. More Selenosmilia hanging down from the ledge. And there's a live Desmophyllum on the right side of the screen of our view.
You can see how we're just about to
Ooh, and what's this?
move up onto the flat part of this ledge.
I'm not sure. Is that a bamboo coral? The thing on the top right? This coral here?
Yep, that's
Yep, it's the same kind of thing. It's grown and then taken a different angle.
Yeah, this one I think is different from the one we just saw to the lower left.
Nice.
I'm saying that based on the thickness of the skeleton and the size of the polyps.
Les Watling says that they've seen this fish on Rehoboth with the Acanthargorgia armata. Which is what? What's the Acanthargorgia armata?
That's an octocoral.
Okay.
Wow.
This is lovely.
Yeah.
And Pilot, can you drop the camera just a little bit down?
Dropping camera.
That's the first of the day. Might be.
Yeah, down to that other large bamboo coral that we were.
Copy this one down left.
Yeah.
Let's get lasers on it real quick.
Scott, is it normal for them to kind of take a big turn in the skeleton there? They start out growing straight out and then grow upwards?
Yes, absolutely.
Oh, wow.
It's very common for many of these octocorals to respond basically to what the local environment is at the time that they're growing. So we'll see, for example, sometimes they may settle on the underside of one of these ledges and grow kind of like a J shape up and out.
Huh, that is really interesting. So they're making sure to maximize their place in the water column and getting all the current that they can for food delivery?
Yeah, and another way I think about it is, when the larva settles, it's very small and is looking for a place where it's not going to be buried in sediment. And so on the underside, maybe more protected, grows enough that once it's of a size, then it curls around and gets into the maximum water flow.
Wow, that's awesome.
So this is a really nice view of this bamboo coral. One of the things we do as taxonomists is we try to get some sense of how spiny it is in terms of the sclerites, the microskeletal calcareous elements. And I'm not really seeing a lot of needles on this one. That'll help us later. This kind of feels to me like it's jasonisis.
Come wide.
Huh. Okay.
And I use that term
I have you
loosely.
at
That is
80,
the name
but
of a
I don't think I'm
genus.
swinging. So.
And for those listening who are familiar with genetics, we build these phylogenetic trees, and there are clades. And this one looks like it belongs to members of a clade that we call the J clade. And we call it the J clade because the only thing that's described there is jasonisis. But we know there's more diversity. It's just it hasn't been described yet.
Copy that.
Wow.
We don't want to spend too much time here because we are starting to fall behind, but if you see it as I rise, just let me know.
Cool shot, isn't it?
Bamboo coral that's on the upper ledge there, I think that's a different one. I'm going to say that's an Isidella based on that side view, and something about, to me, the way the branches are arising looks similar to something that we have been calling in the past Isidella.
So another
but it's certainly different from the one we just imaged.
That's an incredible geologic view, by the way. We look at that.
Biology.
Right. I just want to mention the geology again before we go up on the flat plain, because we have been climbing and climbing, what is it, over 30 meters of relief and going through geologic history. And if you think of what those rocks, the stories that they could tell, if we could get samples out of each one of them or core right down through all of them, they are telling the story of the edge of this continental margin through their sediments. They tell us of the chemistry, the water flow, the microfossils within them, the ones that are benthic organisms. They tell us about the seafloor conditions. The ones that are falling down from the surface waters are telling us climate changes because we know certain species like certain water temperatures. So there's an incredible story to be learned if we could get samples at every portion of that wall that we have climbed. Sadly, we aren't equipped to do that, and it would take far too much time, and we'd miss all this amazing biology. So I'm not whining. I'm just pointing out that it would be great to come back here at some point for the purpose of unraveling the history of this platform.
That's a nice looking coral.
That black coral.
Gosh, that's a huge
Put a
pathies.
couple of different directions there. Did a double.
And the stalk of a bamboo
Single.
in front of us. Looks like it hasn't decided to change its direction yet. Or maybe
Can we
it's
get a look on the white coral that for me is in the upper left, kind of pointing towards us like a plumose feather?
for the upper.
Pilot, this
Might
is
be a
watch
new
one.
black
Go
coral
ahead.
for the day.
Can we, there's a white coral that's now in the upper right of our view. Could we get a zoom on that, please?
Yeah.
Yeah, copy that.
Yeah, that looks like something different than we've seen today.
We'll see if Tina responds. I'm wondering if it's an umbellapathes.
I think it's almost time to turn north. Do you guys agree? Judging by D2 sonar.
Pretty much at waypoint 2.
See that?
Yeah, do we want to run the ridge?
Yeah.
Is that the idea?
I
Yeah.
talked to watch it, but I think it's time to go northbound.
Yeah. How about you can kill lasers? In video, you can come on in.
Yeah, let's.
Wow.
Tina says this would be alternapathes, not umbellapathes, but that would be new for the day as well.
Wow, that's great.
Beautiful.
So, do you want to put
This is another
Sirius
example
right on
of a
top
black
of
coral
P2
that's in
and
the
then head
family
north?
Schizopathidae, and they
I might
was
kinda like
describing
where
this the
you're
other
at.
day. What's really
That way he
interesting
can
is the
kinda
polyps are
ride that
extended
ridge if he wants to.
linearly along
Yeah.
the branch,
he's-
and so it
I'm gonna talk to
appears like
Watson's.
the tentacles come out in
Once
pairs instead
Scott
of in
Francis
rings.
done.
Copy.
Watch
Some of
these.
the
This is
polyps
our
are
ROV
really
Navigator.
swollen, and I don't know if that's
Go ahead,
because they're
Nav.
full of
So we've
the
got to
eggs,
waypoint 2. We're at the
they're in
top.
reproductive state, or if some of them
And
are
it's time for us to
feeding.
turn
Probably
north.
reproductive.
We can either ride the top, or we can kind of ride the top a little to the east and then kind of ride that ridge. What would you like to do? Okay. Yeah, that's what we were thinking too. Yep. Understood.
And we're going to take a look around here, and
you
then
guys
we're
hear that?
going to
Yeah, stay on the
take a
east
turn
edge.
to the north and
Once you guys
follow
acquire that,
the top
we'll
of
get a heading
this,
on that. I think it'll be
the
about northbound.
ledge here,
Yeah, so
and look at
as
the
you start
underhang.
heading north, I'll just follow you and we'll figure
and
out.
sneak
Sounds
peeks
great.
up over the top here too as we go along.
Teamwork turn.
Yeah.
Copy.
And this is pretty much, you can still see the edge in Sirius, so this is pretty much it.
Yeah.
Yeah, that's what I was
This is a good
suggesting.
spot. So you can, that way,
Yeah.
you can kind of hop over the edge or ride the top or
Yeah.
the edge of life. I like that, edge of life, because it kind of like ends. You know?
Do you get lasers? Co-pilot. Kind of run with them. Nice.
I would say that the Southeast Coral Deep Initiative chose a really good spot to dive today.
Oh my goodness, you're so right.
This is going to add a lot of information to the database on the distribution of these corals. This is great.
That's excellent.
I was wondering how
And all the
many hours it's
organisms
going to
associated
take
with
to
the
go
corals
back
is
through the.
Big fish as well, so beautiful spot.
Yeah, how many hours it'll take to go back through and identify everything that we're seeing because there's just such diversity.
And another thing that's kind of fascinating is the contrast with the dive we had that wasn't too much deeper on the northern part of this Blake Ridge. But it says a lot for the geology, right, Leslie? We weren't seeing this much exposed rock and edges on that other dive, so you have to have these right
So
conditions for
I'm
all
just
these
gonna
organisms
turn a little bit
to
and
find
kind
a
of
place
see
to live and
what's
to
the
grow really well.
heading of
Yeah.
the ridge. Okay.
Sounds good.
The general gradient of the northern portion of the Blake Escarpment is
It's probably
much
about
lower.
there.
As you move farther south,
That's like
even of this
010.
site, it becomes nearly vertical.
So
So we're in that transition.
So what we can do, Fernando, is have You
Oh,
can,
it looks like a
you
basket
can
star
look,
on the top of
Yeah.
this large
like,
bamboo
a
I'll
little
be
bit
lined
to the
up
right?
left.
340, but
And I'll
we'll
move.
Good observation, Cheryl. I don't
So it's
see
about
very many of
010.
It was actually Annie.
We have
that I-
Annie, our videographer here, who's seen a lot more of this than we have. So she's been learning along as well. So we're at a depth of about 1247,
Do you want me to start moving
1,247
right now?
meters.
Or wait a minute.
We
You can go
began
for
the
it.
dive at over 1,300 meters, so we
Bridge, this is
it's
RV
about
now.
an 80-meter ascent up this wall.
Bridge.
Yeah, we'd like to move 20 meters.
And
bearing
parts of it were
010
nearly vertical.
degrees,
Well, they were
speed
vertical.
decimal 2.
And the rest was about 40 to 50 overall this slope
Roger, I
from
understand. 20
top to
meters at a
base.
bearing of
We had estimated from the
0.10 at a
maps
speed of decimal 2.
at being
good
about
copy.
30 degrees. So sometimes with the multi-beam
Can
sonar,
you
even
zoom out a
that
little bit?
high quality,
So
high
where's
resolution data, you can't always
Directly north here.
pick out the
Okay.
changes,
All right.
subtle changes
I ride this ridge.
in gradient.
What time is off bottom?
3:30, 3:23.
Let's pass this shift, correct?
Yep.
We are learning
done at
so
2:30.
much more
And then
through
how
the
far is waypoint
mapping
3?
that we're able to do now. We're able to pinpoint areas of high relief like this.
I got
That in the past, the Google Earth map base
ROV
that you
nav,
see is a
bridge,
very
move
low
initiated.
resolution,
Move
very
initiated, take the
large
bridge.
pixels. So they
I
miss
got
all the fine detail
120
features.
meters there. And then after that, we can go towards
Now we're
1.5.
looking off the edge of that
is
wall that we
300
just
meters.
climbed.
The top of
We
this.
have a look at the fan that's just to the left of the edge of that wall, the whitish one. Might be something.
Right in the center.
Pilot, this is watch 1.
Go ahead.
Could we get
Probably in your
a
lower
snap
right now.
view of this coral that's just to the right of the lasers? And actually, the one that's at the lower left would be good also if it's possible, but if not, just at least the one on the right.
Yeah, copy that. Video, go for snap. Nice, Kappa.
Looks like the ship started moving, guys.
Copy that.
Okay.
Thank you. This is the one that I meant. Definitely another bamboo coral. I'm just not convinced it's the same as the ones we've been seeing. Very nice. I see some nodal branching there, so that'll be very helpful. And just if you look at the upper part, there's something swollen. There may be some overgrowth in this colony, or some kind of maybe an egg case or an anemone, or maybe even something parasitic.
And where was that you saw the swollen area, Scott?
I'm sorry, can you repeat that, Cheryl? I didn't hear you.
Where was that swollen area that you noticed?
Oh, it's near the top of the colony on the left side,
Okay.
Left side.
center left. I see we have Tim Shank in the chat room today, and Tim is really interested in all these associates that live on these corals and sponges. Oh,
Do you
there
want
it is.
to come out a
So it's
little
just a
bit?
cluster of branches. I don't know that there's anything
Not
in
quite
there, but there's that
seeing.
odd branching. We passed over it, and I didn't actually see an
Oh, yeah.
associate. But very often we see that if there's been some damage to the colony, it has the capacity to
Did I pass it?
regrow and heal itself. And often when that happens, we get this very odd branching pattern. So that's what's going on.
Okay. And thanks for joining us, Tim. We were seeing some Europtychus on, I think it was Paraanthropathes.
Yeah,
Just about
copy.
everyone we saw, but that has been the most common associate that we've seen so far.
Do you want me to go back in, watch lead, or should I move to the other? Copy. That's right. My co-pilots get lasers on this one real quick. Great. You kill him?
So is that a little Liopathes to the right of this bamboo coral?
This one seems to have somebody living on the upper left.
Yeah, it might
It's
have
reddish.
an associate.
All right, video, let's go in.
ROV nav, bridge, move complete.
Move
Yep,
complete.
looks like
Thanks,
an
Bridge.
ophiuroid.
And it looks like it's had, well, we'll look at it in a minute. Looks like it's had its arms nibbled off recently and it's regenerating. Unless that's characteristic of this species. Maybe someone can weigh in on that, because it looks like all the arms are that way. I've never seen a brittle star that has such variation in an arm.
But one thing you can see here really nicely illustrated is where the brittle star is moving its arms. It's kind of disturbing and tickling the coral polyps. And so you can see all the tentacles retracted compared to on either side where the tentacles are still extended. And so it looks really fuzzy when the polyps are happy and out in kind of a fetal position. And then the ones where the tentacles are retracted, you can see these little spines arising, which are these sclerites that we think may have somewhat of a defensive function. I don't know that this brittle star is feeding on the coral. I don't think it is, but it's at least disturbing those polyps.
It's bothering it. Yeah, and what I was thinking about the arms, one of the arms does not have that white extension, so you can see the one right now on the upper left. And so I think those might be the regrowth.
Yeah, I think there's a thought that fish will come along and nibble the ends of the arms of these sea stars.
No wonder it's trying to get some cover.
Yeah, and Tim Schenk agreed with that. It's probably regrowth.
If only we had that ability. Look at
Just took
that.
a big leap. We have some Selenosmilia, mostly dead, a colony to the left of the bamboo coral that we've been looking at. Several glass sponges.
Do any more views, Watch Leads? It's about time to take off.
Okay.
Yeah, I guess we can move on. We could look here forever, but we don't have that much time, so.
Sounds good. All right, Copilot, you want to grab lasers on?
Any idea, biologists, what the little white stalks are to the right of this coral in the front?
My guess is that they're probably polychaete worm tubes.
Huh. They're so white, I'm surprised.
Anybody else have another opinion?
Well, another guess without looking closely is a sponge or I seem to recall there was something like this that was little hydrocorals or something.
It looks
I'd
really
have to dig back at
greenish
some of the
on
previous
your
dives.
screen. On mine looks...
My memory isn't good enough.
Huh.
Is it greenish?
No,
Was
I
that
think
a
it was before.
Metallogorgia
It was
there
the lighting.
in the top center?
Oh, yeah, look how beautiful that is.
Pilot, this is watch one. Could we zoom on that coral right in the center there?
Yep. All right,
And it
Copilot.
would appear...
Definitely looks like a Metallogorgia from here with its characteristic
And
associate of the
video can go in for zoom.
Ophiocrius oedipus, which is a long name for the brittle star that is a very close associate of this coral. Everywhere, this coral is very widely distributed throughout the world. We find it in all oceans. And every time we see one, we see one brittle star in the branches.
Oh.
And it always seems to be this one species of brittle star, and we never find this brittle star anywhere else. And even the smallest of these Metallogorgia colonies we see have these brittle stars, which suggests there's a very close association, and it leads to a whole lot of interesting scientific questions. For example, how does the larvae of the brittle star find that really small coral? And, you know, because we never see any of these corals without the brittle star, there's some suggestion that maybe it can't survive without the brittle star. And so that association is really important. And maybe the young colonies that don't find a brittle star don't grow. You know, I'm just kind of making conjectures at this point, but it's such an interesting observation that we see throughout the world that there's clearly something going on.
That's definitely one of the big mysteries with these associates, is how they find their host and how important the relationship is for both of the species. It's just amazing.
A follow-up to that, we have on occasion seen Large Metallogorgia colonies that have died, I don't know if that's old age or something else, that still have the brittle star on them. And that suggests that the brittle star can survive for some period of time after the coral has died. However, earlier today we saw, both Cheryl and I commented that we thought we saw a Metallogorgia skeleton completely dead and overgrown, and it had no brittle star. So after some period of time, the brittle star either dies or
You said small fish left? Watch it.
Scott, are Metallogorgia known from this region?
Yes, they are. We have collected them just to our south in the Bahamas and the New England seamount. And I think we saw them during the 2014, I guess, cruises in the canyons to the northeast. And we were actually quite surprised during those Okeanos Explorer expeditions in 2014. That was the first time we'd seen them in the canyons. So this may be a first observation for this particular area because we haven't had dives this particular part of the Blake Ridge or Blake Escarpment, but they are known to the south and north and offshore.
The smaller ones or the larger ones?
Thanks, Scott.
Copy.
So would this be a Liopathes species here?
Alright, Kapot. Grab those lasers. And video, you can come on in.
Yes, 2 Liopathes, another black coral species. This one grows in kind of a more bushy type of growth form and very, very delicate branches.
Yeah, one of the stories I can share with you about Liopathes is that it's one of the champions at producing mucus. We have collected samples, parts of branches before, and when you put them into a bucket, they can literally produce, pints of mucus in a period of an hour. Quite amazing.
Quite amazing for such a delicate-looking species, especially.
Yeah, the stalks or the skeleton is more arced. It's not straight growing like the ones we've been seeing.
Yeah, another interesting thing about Liopathes compared to some of the other black corals is the degree of spinous or thorniness. Black corals also have a common name, thorny corals, because the skeleton has a lot of spines growing out of it. And the shape and distribution of the spines themselves is actually used in the taxonomy of black corals. But when you look at the thicker branches, the main axis, the older branches of the Liopathes, they're really smooth. They're beautifully smooth. And so it seems like the spines become overgrown as new layers of skeletons are laid down in the older parts of the colony.
All right, are you happy with that, Washlet? Leads?
Yes, sir. That's great. Thank you.
Great. All right, video. Let's keep going.
So again, we're traveling across the edge of a very flat, topped, and then very steep sloped ridge-like area. It's part of a larger scale of terraces that step down from the Blake Plateau.
Wait a minute. What do you think?
Toward the Blake Ridge.
Hey, push real quick if you want to get one going.
Yeah, sure.
We're just doing 20 meters, or?
Yeah.
Yeah.
Bridge, this is ROV nav.
Bridge over.
Pilot, this is Watch
Yeah,
One.
they can move, same
Yeah,
as
go ahead.
last, 20 meters,
Can we
bearing
move just
010,
off of this,
speed
ledge
decimal 2.
so that we can
Okay,
take a
20
look at
meters,
the
bearing 010,
underhang
speed decimal 2.
of the edge of this plateau here.
Sorry, you said move away from the edge?
Move along it, so not right on top.
Oh, so keep it in
but
view?
Yeah, a little over the edge, so we're over the water column, but we're able to look under the actual edge here.
Yep, I can definitely do that.
Ooh, I think I see a big one.
There's another one of the sea stars. We've seen a few of these at this point.
Do you want just like this, Washlet? Or do you want me?
Yeah, or maybe a little further out so we can peek underneath. There's
Yep.
a lot of
ROV nav bridge, move
just
initiated.
underneath.
Move initiated. Thanks, Bridge.
I'm going to grab lasers. Co-pilot.
Admit it. You just want to see the geology.
I do.
Okay, good.
Yeah, there's more live Selanismilia. There's a little fish that's sticking out. Maybe, oh no, maybe that's not a fish.
Oh, I think it's one of those sponges that has
Yeah.
a big
It
hole
looked like
in
an
it.
eye.
Yeah, it does look like one.
Another glass sponge, some more Selanismilia. So this habitat reminds me of the canyons up in the New England area and the minor canyons that we visited last year in the Henry Bigelow cruise. And that lip at the top of the scarp was often where a lot of corals were. And you know, if they're able to get underneath something like this, they're protected from sedimentation. So
Sure.
it's quite a prime habitat, I imagine.
And those overhangs provide great habitat for fish. So we've seen one beautiful fish. What was the red one? Recollection. I don't know if we ever had that identified, but we got some nice video of it. And we'll be keeping an eye out for more species.
You still like that bearing, Fernando, or do you want more north next time?
I feel it's fine.
Okay.
Yeah.
Oh yeah, now I can kind of see.
It's another black coral
Yeah, if
coming
we
up
start
here on
getting
the right.
too close to the ridge, I'll let you know, and we'll.
Okay. Yeah, it seems to be a pretty good situation. Sometimes I forget that like your heading isn't where we're moving, you know?
Yep.
It's a little, yeah.
Yeah, this is Copilot and Navigation 202.
Yep.
No,
Next
101, no, 201.
Next level, yeah.
Glad we were looking. Very nice.
Wow.
Nicely done, pilot.
Don't want to bump that one. Looks like it's like ready to fall
Another
off.
very large bamboo coral coming up here.
The crinoid to the left.
That one must be, goodness, 3 feet tall at least, I imagine.
So that rock must not be that friable. There's no way something of that size in a current
Right.
could hold on to rock that was going to break away.
Let me know when you want me to turn off lasers.
Copy that.
I'm gonna try this still cam on this one. Don't have to, obviously, but
It didn't look good on the other one. I think like colors and I don't know.
I think
The other colors look, the green
The
ones
awesome
look
majesty
better.
of
We
the
can try
bamboo
it if you
coral.
want.
no, it's
That's for sure.
Quoting Scott France, and we totally agree.
What do you say, Sean? You have to.
Especially when you think
Whatever
how
you guys
long it
want,
took to
yeah.
get to this
Just
point.
flying along.
got it
That's
there. Might as well use
beautiful.
it, right?
Yep.
Might as well try.
All right.
Yeah, pilot, I think if we fly along very close to the top and, you know, zoom out, zoom in.
Are you
we'll
okay
get
if
a
I turn
sense
on the
of
fog lights video?
Finding new
Sounds
new
good.
varieties and abundances.
Sounds good, Washley.
All right. So the still cam's on. Levi, let Sean know
We
where's the
have
best picture.
More Selanismilia, stony coral, just to the right of this large
I think
bamboo.
we gotta pull the camera out and change like the ISO or something.
I don't see any
The bottom light's on?
associates, at least large associates on that. Maybe,
Yeah, go for zoom.
Oh, there might be
maybe
brittle
up on
star.
the, yeah, brittle star and the upper stalks toward the back.
Stars.
Yeah. There's
So is
only-
that an accurate representation of the camera?
Yeah. That's the picture it's taking.
Huh.
I think there must be like some sort of adjustment.
I mean,
or something.
when Dan downloads them to his laptop, he color corrects them.
Pilot,
and
maybe on
they
those
look really
back
nice.
stalks.
So
Back
Yep, say right in the
stars.
middle there's
Yeah, I
a,
see
yeah,
'em.
you see them.
no, they will look really
Back
nice.
video.
It's like somebody nibbled on one of those stalks too.
Yeah, the polyps are missing
I
Go
think
for
it's
zoom
too
video.
dark.
a portion of one branch.
Get 'em.
See.
Yeah, that could be the culprit there. He might be
Look at
munching
that.
on
He
those.
is definitely,
Munching on
well,
it.
not definitely munching, but he's definitely on a dead portion.
That's a really good observation. As I said earlier, I'm not used to seeing these things feed on it, and we don't know that it's feeding, of course. It may be that it's just abraded the skeleton so much, or the tissue so much with those really spiny legs that it's worn the tissue away, and now it has a nice perch to hang on to. Interesting.
Do these tiny polyps, I think you told me yesterday that all of these corals have nematocysts, the stinging cells.
Did you get what you'd like for this one?
Perhaps it's seeking areas that don't have the polyps because it's being attacked.
Well, so interestingly enough, the work that has been done on octocorals show that they don't have much in the way of nematocysts
Oh, they don't.
compared to all the other
I'm
cnidarians.
gonna turn off the fog lights
Or
video.
if they do, they don't seem to be very numerous
Let's turn
or
off
very
the still
potent.
cam.
So that's still an open question in the world of
Just Fernando, you've
deep sea
seen
octocorals.
very dark photos like that being taken, and then they were able to brighten them up, or this is
We
darker than...
we
white
didn't take pictures of
octocoral that we're
corals
coming,
like this.
upon now is
We only
another
took
bamboo, I
pictures
believe.
of. Remember the first one with the green sponge?
Yeah.
That one?
Yep, I
you
agree.
could see it there,
looking
and
at
it
that
looked
last
better.
one, I had made a note in the chat
FRV
log that
nav
that
bridge
may be a
position move complete.
keratoisis
Move
D2
complete.
plate,
Thanks. You got
but
us to do with
the
the
tissue
color
was
thing?
so thick it was too
I
hard
don't
for me to
know.
see
Work
where
with
the
Dan
branches
later
were
today and see
arising,
how they look.
nodal or internodal.
Maybe
Yeah, it was
If
a
it
lot
was
of
a
tissue.
that view
keratoisis,
is deceiving.
which I'm
Maybe
sticking
it's gonna look great.
Because
it would
remember
be
when we
an
were on the
internodal
shelf down
branch.
low?
Another great
everything's
sponge.
stuck up
Really
there.
nice diversity
Right?
for all kinds of things.
Yeah,
but
maybe
certainly
this
the
camera
bamboo corals
catches
today.
And it
the
seems
greens
as if
better,
the bamboo
but maybe
corals
the
are
actual
quite
image will
numerous
look
up at the top
just as
here,
good.
but
Who
we're
knows?
few and far between as we were lower on this scarp.
That's a nice looking sponge.
Yeah, good observation, Cheryl, and I think that is consistent with things that we've seen in the past. They don't do so well on the sharp face of the wall itself, certainly lower down,
A little bit of a
probably
heavy
because
pilot.
those pieces will break off more easily.
Just
Although we
hard
did
after
see
a
a
while.
few, right, that were sort of growing out of the wall
Make sure
and curled
you
up.
don't fall
So they
asleep.
must have found a stable
We
spot.
gotta keep you awake.
But they certainly do seem to like being up near the edge or just in from the edge of these escarpment-like features.
And we have a big, beautiful sponge coming up here. This looks different than anything we've seen before, I think.
Video, try to get.
And to its right looks like another of the Venus flytrap anemones, perhaps.
Yeah.
We saw them lower in this dive, lower on the ledge here, and it was pink there. This one's a little darker in color.
Could be that we're looking at the side or
Yes, yeah.
underside of it. Oh, and there's an anthomastus growing off the back of this sponge. Wow, that's a gorgeous
Kill
sponge.
laser strip that.
Laser soft.
And there's some zoanthids that are growing on it also, on the sponge on the edge that was pointing down.
It's like a vase almost.
Yeah.
There they are, zoanthids.
Looks like there's a dead fish, in the, or maybe a living fish, but it looks like fins in the base of the sponge.
Base?
You see the fins to the left side of it? Up here.
Yeah, I was thinking that might be a piece of seagrass or something. I'm not sure.
Yeah, I can't tell. Or maybe it's something living under it. It's hard to tell. Just can't. Yeah, it doesn't look like a fish. I was just hopeful.
Yeah. We haven't seen all that many fish considering the diversity of other things we've seen. Oh, it's a hermit crab on the lower left of the sponge, and then the anthomastus octocoral that's growing on the back.
I love the beard of the coral on the
Yeah, zoanthid beard.
zoanthid beard, yep.
And again, more
Alright.
Selenosmilia stony coral on the left.
So we made some observations in the Pacific of a specific type of sponge that also, every time we found it, had
Thanks,
these
Pilot.
zoanthids growing from it. And zoanthids are colonial anemones. They're known to associate with different kinds of invertebrates, including sponges. But I haven't seen one that looks quite like this with the zoanthids. But it's a really interesting observation.
Yeah. Here's There's the flytrap anemone.
Do you want zooms
Looks
on
like he's
flytrap?
inhabiting
That'd be
it.
great. Yeah, see if we can.
All right, video go for zoom.
Just a quick zoom.
Probably a stalk of a what is now a dead octocoral.
I think we're looking straight down into it, right? So it's missing some tentacles on the left. Or are they just pulled back?
The mouth is over to the right of what we're seeing, and this is the side. Is what
Oh,
I'm
I
thinking.
see. I'm thinking that's a concave surface. It's probably convex.
Yeah, I think that's
Yeah,
right.
it's optical illusion for me.
Yeah, we're both leaning over.
We're trying to see around it.
It looks like you could reach out and touch it.
Yeah. Pluck it off the rock.
But instead, it's 1,249 meters below us.
I see. So there's the mouth.
It's kind of folded a little
Yeah,
bit.
yeah. Feed me. Maybe he'll open up for us. Maybe he thinks we're food. Let's get a good shot of the stalk there. Oh, that's a beautiful picture.
All right, video, let's come wide.
The edge of each one of these terraces seems like a good place for corals, and then any of the flat places also. It's just an amazing coral dive.
All right, what's ship status, Nav?
Ship is stationary. Ready for a move?
How do you feel, Copilot?
I feel good. Whenever, if you're gonna keep going, yeah, let's get on moving.
Let's do it.
Still happy with 010?
Yeah.
Okay. Bridge, this is RV Nav.
RV Nav bridge.
Yeah, I'd like a move same as last. 20 meters, bearing 010 degrees, speed decimal 2.
RV Nav bridge, 20 meters, bearing 010 at decimal 2 knots. Over.
Oh, and Andrew
good copy.
Petrini added in the chat room about IDing that pink fish that we saw earlier, Gadiropsaurus, and she thinks it could be a range extension.
Oh, that'd be great.
Seems pretty far south for
Is
this
that-
genus,
Oh, we missed
but
it.
she's checking the literature.
Can you grab lasers real quick, Copilot? Nice.
RV Nav bridge, move initiated. Over.
Move initiated. Thanks, Bridge.
So as Cheryl noted earlier, all of what we're seeing now, it looks like almost root systems, but it's really dead coral. It's the scleractinian coral that we are seeing here and there as living organisms, but this is the dead remaining skeleton parts of those corals. It's called, let me try it again, Selenosmelia.
Yes, good job.
Okay.
Wow, there's a lot of it here.
Yeah, and the reason it's very dark is because of being encrusted by the oxides in the seawater precipitating out of solution onto them and giving them a coat. And those are manganese and iron oxides in this area, very common. Even in the sediments, we'll see the fossilized planktonic foraminifera. Some of them are bright white as they are in life.
No, they were talking
And
to...
many of them, most of them are actually
Copy.
rusty color because they've been on the seafloor long enough to get a little very fine coating of that iron oxide. We have another of those
Ooh, a Metallogorgia?
Metallogorgia with its associate. It's just going off the screen in the bottom. Oh, and what is this? I almost
Pilot, this is watch one.
wanted to
Yeah, go for
say
Could we get a zoom on the coral that's just in the
the
lasers now?
coral, yeah.
Yeah.
We
Video.
saw one
Thanks
of
very
these
much.
earlier.
Go for zoom.
I think of these as fireworks. They look like fireworks
They
to
do.
me.
And it looks like he has this ophiuroid
Wow.
associate.
Is that just
4th of
one?
July
It
under
looks
the
like there
sea.
could be more than
It
one.
must be
That's-
more than one, yeah.